together. And I specifically just lift up any of those that were in Tennessee, Georgia, Alabama, Mississippi, I think maybe even Louisiana, that were affected by the severe storms yesterday. Just pray that you be with anybody that has lost significantly. I just pray that you be with my students that are struggling to figure out how to do online classes with spotty to no internet. And just pray that people get the help that they need as quickly and safely as possible. I also lift up Katie's dad, Chip, as he gets ready to have a biopsy on Friday, and just ask for answers and solutions for him after Friday's appointment. And as always, I continue to lift up any unspoken prayer requests that are on these students' hearts. I thank you for loving them and hearing them. It's in your son's name that we pray. Amen. All right. So let's get started. I feel for any of you that are in this area that are probably super tired like I am because they did not sleep much last night. So just bear with me, do your best to pay attention and stay engaged. All right. Desiree, do you have your fact?
I do. There's a condition called broken heart syndrome. It's believed that if there's like enough stress, it'll cause the heart to enlarge in size. And it's said to, I guess, affect the left side of the heart, the left chamber of the heart. Like it constricts the veins and stuff.
Interesting. Hannah, is that what happened to Grumpy Cat? Is she listening?
What?
Isn't that a picture of Grumpy Cat?
Yeah, I think.
You think? Perfect. All right. Thanks, Desiree. Let's get started with the immune system. Okay. And remember, if you haven't said hello, go ahead and say hello just so I have proof of your presence. Okay. And also, I think if you guys have seen any of Dr. Khan's meetings, you are probably aware of the I guess emergency grading policy. And so today's a little bit of an exception. So one of the things that works with that policy is that there's an attendance requirement set by the professor. We don't really have that many classes left, and so my attendance policy will be 100% of classes being attended in order for you to use that pass-fail policy if you choose. But, right, in light of recent storms and events, I'm not going to start enforcing that until Wednesday, depending on how people's internet and things are going, maybe not even till Friday, right? And so I know there's a couple of people gone that contacted me that either have spotty internet or no internet right now because of the storms, and so that's totally fine. Don't freak out about that. And I'll send out the document that we got that maybe you guys have seen, but essentially what it's going to be is if you have a D or above, you could choose to have a pass, a P, instead of your letter grade. All right, an F is still an F. And so, situations where, I'd say, well, if you have an A or a B, I would probably take the letter grade instead of the P, right? But that will ultimately be up to you, and you will be allowed to make that decision after you see your final grade. All right. So just in case some of you had been kind of following that, we're keeping up with that. All right. So let's get started here. If you guys remember, we had started talking about, or had been talking about antigens and epitopes. And I know we did this, but I just wanted to do a quick review, right? An antigen is either a whole pathogen or a part of a pathogen that is recognized by the immune system. Okay? In this case, we're looking at antibodies, and antibodies are going to bind to specific areas of that antigen, and those specific areas are called epitopes, right? So here it is showing you one antigen that's being recognized at 3 different epitopes by 3 different antibodies. And remember, this a little bit of repetitiveness is really essential to ensuring that the immune system is able to properly get rid of and dispose of any type of threat. Okay. Ooh, and Kelsey, can you remind me what cell produces antibodies? Kelsey, are you there?
Can
Oh,
I just guess? Yeah, sorry, I don't know.
you don't
Can I
know? Hey, what's the grade that comes after an A?
A B
Ah, there we go. B cells. Perfect. Nailed it, Kelsey. See,
scored. Thank you. Thank you.
that's so impressive. All right. So for the immune system, right, when we talk about, you see here, antigen recognition by lymphocytes. Remember, at this point, we are in the adaptive, immune system, right? You could also call it specific immune system if you want. And so we've mentioned these, right? And Kelsey just pointed out, right, the B cell, right? But we also have T cells that are really important. So these are the 2 main players in the adaptive immune system. Okay? And you can see that these B cells and T cells are loaded full of identical antigen receptors. And that's actually a really important point, because when we say identical antigen receptors, I think I've mentioned this before, but you produce B cells and you produce T cells that are very specific for a certain pathogen. Not only are they specific for a certain pathogen, they may be specific, or they are specific for a certain epitope on that pathogen, right? So on the last slide over here, let's see, I can do that, where we have 3 antibodies that are recognizing 3 different epitopes. Although these antibodies are fighting the same antigen or the same pathogen, right? However you want to look at that, they are being produced by 3 different B cells. Okay? So that's what we mean with identical. So this B cell here that's recognizing or producing antibody A, it has receptors all over itself that resemble antibody A. The other B cell has receptors all over itself, antibody B, and so on and so on. All right. So that's what we said right here. All right. And then again, each lymphocyte, displaying specificity for a particular antidote. Right, so everything just said there. So B cell receptors. All right. B cells are kind of interesting because a B cell receptor actually looks exactly like an antibody, right? The only difference is a B cell receptor is not secreted like antibodies are. And so for a B cell, right, to do its thing, an antigen has to bind to a B cell receptor that then activates the B cell, which we may go into a little more detail. But after that B cell is activated, it then secretes a bunch of almost identical receptors that are then secreted, and that's actually what antibodies are. Okay, so that's what that's telling you right here. And please, let me know if you have any questions. Right, I'll try to keep an eye on chat, and you can just speak up if something catches your attention that you need clarification on. So this is a diagram that shows you what a B cell receptor on your left and a T cell receptor on your right looks like. And you can see that they are pretty different, but they also share a lot of similarities. And so you see the B cell receptor here looks exactly like an antibody. It has that nice Y shape. A couple important areas on it, right? And you'll see similar areas on the T cell receptor. Here you have variable regions represented by the V, right? So variable is important because this is what gives this receptor specificity and allows it to bind to a specific epitope of an antigen or pathogen, right? You see here that you have these constant regions. These constant regions are going to be the same no matter what the antibody binds to or what it recognizes. Okay? You can go over here to the T cell receptor and you can see it does not have that same Y shape, but it does have similar regions. So here you have variable regions that are looking for a specific epitope on an antigen or pathogen. And then here you have constant regions. So one of the things, and we're probably not going to talk about this in a ton of detail, that I do want to point out, it's important, is B cells are able to bind free floating antigen. All right. And so what that means is if there is a pathogen or pieces of a pathogen that are floating around within your body, the B cell receptor can bind to them and interact with them. All right. This is actually pretty different than a T cell receptor. A T cell receptor actually has to have antigens presented to them by another cell. It can't recognize free floating antigen. Okay. So basically B cells can go out and grab whatever is going to bind to them. T cells have to have it handed to them. All right. So we just talked about the alpha and the beta chains, right? You can go back here and you can see those, right? Having those variable regions and those constant regions. Okay. So this is kind of just setting up, or I guess affirming maybe what I just said. But I just mentioned, right? T cells can't bind free floating antigens. So they have to have something presented to them. And that something happens to be MHC molecules. You may have also heard of these called HLA molecules. MHC molecules or HLA molecules, right? Same thing, are actually one of the things, if you've known someone that's had an organ transplant, this is one of the things that they're actually looking for similarities between the donor and the recipient. The more similar they are, the better chance that that organ will be accepted by the body. The more different they are, then the greater chance that that organ will be rejected. All right. And we can talk about that a little bit more later. All right. But different cell types are going to have these MHC molecules, which then present the antigen to the T cell receptor. And then it just tells you down here, MHC, in case you're wondering what it stands for, major histocompatibility complex. So, one thing I want to point out here, and this is a good point, right? And it says infected cells produce MHC molecules, right? And those MHC molecules contain small fragments or portions of that antigen from the pathogen that's causing the infection. They're then sent to the surface where they can be recognized by T cells. Okay. It's also possible and totally normal, though, for MHC molecules to present normal antigens on them. All right. One of the things that maybe you guys have heard about before in biology, but if not, hear it now, right, is that your cells, even when they are healthy, are constantly undergoing remodeling. And part of cell remodeling is actually degrading old proteins, right? And sometimes when those old proteins are degraded, they are also presented on MHC molecules. But, right, a T cell that is doing its job should be able to determine or distinguish self, right? Those normal proteins that are part of the remodeling process that have been broken down from non-self as a pathogen. All right, so MHC molecules can present both pathogenic antigens and they can present normal protein components from a cell that's being remodeled. Okay. Now, here we'll kind of get into this depending on their source, right? Peptide antigens are handled by different classes of MHC molecules. And basically, what this is going to show us here in just a second is that you have something called MHC1 and you have something called MHC2, right? And MHC1 and MHC2 have identical functions, but are presenting antigens to different cells. And these antigens are actually coming from different areas. Okay, so what do I mean by that? All right, well, let's talk about MHC class 1 first. So any cell, right, and for me, you can say any cell that has a nucleus, right, is going to have MHC1 on it, right? Any cell in your body, MHC1 is present. Okay. And this is kind of a good example of or a good example of something being presented that may not necessarily be bad, right? Because every cell has MHC1, and so if your cell's undergoing remodeling and breaking things down, right, some of your own proteins could be presented on MHC1. Right? It's then the job of the cytotoxic T cell, right? This is also called the CD8 T cell. It's its job to recognize, is that part of the host? Is that normal? Or is it something that's pathogenic and we need to be concerned about? All right. So this is kind of a diagram showing you the difference between MHC1 and MHC2. So let's focus on MHC1 because that's what we're just talking about. It's also important to note that MHC1, right? Because remember I told you it could be proteins from the inside of your cell. And that also has to do with pathogen. So MHC1 is typically presenting either something from the self or something from a viral infection, right? Viral infections are taking place inside of the cell, okay? So the way I like to look at MHC1 is I like to think of it kind of as a window, right, that allows cytotoxic T cells to kind of peek inside the cell, right? And when they peek inside, they may see that everything's totally normal and they move on. But they could peek inside and they could see something like a viral infection in there. And then that kind of springs them to action, right? So it's safe to assume at the moment, right, if you're looking at this diagram, right, they're telling you, hey, this cell's infected. And here are antigen fragments, right? This might be individual components of a virus that hasn't been assembled yet. They get loaded onto MHC1, which goes to the surface of the cell. It presents that pathogenic antigen to a T cell. And in this case, right, what would happen is this T cell actually delivers something called a kill package. It releases a chemical signal that actually causes this cell to die. And when this cell dies, it can no longer aid or help the virus, and you stop the spread of the infection. Okay? Now, in contrast, and I actually really do like this diagram, even though it's a little simplistic, but it highlights a couple key points. All right. One, MHC2, and we'll talk about this some more, I'll be sure to mention it, is going to be on cells that are phagocytic. Let's see, pull this up. Desiree, can you remind me what phagocytic means?
Those are phagocytes, right?
Phagocytes, and what do phagocytes do?
They consume pathogens, right?
Consume pathogens, or they can consume cellular debris, right? Phagocytes eat things, right? So MHC2 is going to be present on those cells of the immune system that eat things, right? So we talked about monocytes, which become macrophages, right? Another type that we didn't really talk about much is something called the dendritic cell. Neutrophils can do it. It's not as common in them, but they can also do it. And then to kind of throw you a little bit of a curveball, B cells do it also, but it's a little different, right? But essentially, any type of cell that can eat something or take in foreign antigen, right, has MHC2. MHC2 is not present on an epithelial cell in your lung. MHC2 is not present on a muscle cell in your heart, right? It's not present on a neuron. MHC1 is present on all of those cells. MHC2 is only present on cells of the immune system that are eating things. Okay, who's this? Let's see. Ah, no worries, Grace. Just jump on in. All right. The other, right, kind of an important thing to highlight here is that MHC1, right, is presenting to cytotoxic T cells. MHC2 is presenting to helper T cells, right? Cytotoxic T cells, right, I just mentioned, they kill that host cell that's infected. Helper T cells, they don't kill the cell that's presenting to them because it's a cell of the immune system that is trying to do its job. So helper T cells are going to release signals that aid other cells in the immune system by activating them, by directing them to the infection site. All right, so helper T cells are kind of the linchpin, right? They give orders. They tell you when you should be activated, what you should go after, and what you should kill. Okay, and T helper cells can, right, help control cytotoxic T cells. T helper cells are important for activating macrophages, right? One of those phagocytes to help them be more effective at doing their job. T helper cells also activate B cells. All right, so they are kind of a linchpin, or those of you that like biology of the environmental nature, right? If you've heard of a keystone species, right? So a keystone species, when you remove it from a habitat or an environment, everything else falls apart, right? It crumbles, can't function without that keystone species. Helper T cells are a keystone cell in the immune system. If you remove them, the immune system cannot do its job. That includes the innate immune system and the adaptive immune system. So I think I've already mentioned this before, but I'll mention it again. If you, right, know someone with HIV, if you have HIV, if you've ever read about HIV, if you've heard of it, right, HIV is so devastating to patients later in life because it actually kills these helper T cells. And when these helper T cells are removed from a person's system, right, they succumb to infections that we would commonly be able to fight on our own or easily with the assistance of some simple medications. Okay. So we just talked about this, right, because I had that diagram and I use it to explain that. But, right, MHC class 2, dendritic cells, macrophages, B cells. I did mention you can find them on neutrophils, although it's not as common for neutrophils to be presenting to T cells, right? They display the helper T cells, and, right, I've already mentioned this to you too. MHC molecules, major histocompatibility complex, are really important for skin graft rejection, right, and organ rejection, right? So during a transplant situation, we try to match up MHC molecules as best we can. Again, with modern medication, this is not quite as essential or stringent as it once was, but it still ensures that organ would be accepted by that person for a long stretch of time. So again, we've kind of pointed this out a little bit with the innate immune system. When we talk about lymphocytes that are part of the adaptive immune system, they are arising, right, in the bone marrow. Okay. And again, they come from that lymphoid lineage, And so early on, these lymphoids, right, could develop into B cells or T cells. So if you took one of these lymphoid progenitor cells, it's undifferentiated, right? It hasn't decided if it's becoming a B cell or a T cell yet, right? But depending on signals that they receive and where they mature, right, that determines what type of cell they are. So specifically, some of these cells will go to the thymus, where they will finish their maturation process, and these will become T cells, right? So think thymus, think T cell, right? Some of these guys will stay behind in the bone marrow, and they are what eventually become B cells, right? If you've heard of leukemia, right? And I think we actually mentioned this in the cardiovascular unit, right? Part of the problems, or one of the problems with leukemia, is that you are producing too many of these cells from these stem cells, right? So you're getting too many lymphocytes, or even, right, those other cells of the innate immune system that we talked about from these stem cells. And when you start producing too many of these, some of these stem cells you want to become red blood cells, and the body, right, suffers greatly, because although lymphocytes are important, right, red blood cells are also very important. So if you're producing one over the other, then you have some pretty serious issues. All right. So this is just pointing out, right, B cells start off as a lymphoid progenitor cell. They stay in the bone marrow, they become B cells. T cells, right, will leave the bone marrow, go to the thymus where they finish maturation, become T cells. But then you can see that both can enter circulation, hang out within the lymphatic system and the lymphatic tissues. Okay. And some of these tissues, right, we talked about lymph nodes briefly. There's also your spleen. We're not going to really get into it here, but those of you that like cool intestinal disorders, you have little things called Peyer's patches in your ileum, which are basically little clusters of lymphatic tissue. And so you'll kind of find that scattered throughout your body. Okay. We're not going to go into how they actually regenerate, or we're not going to actually go into how B cells and T cells arrange their different receptors in almost an unlimited combination of possibilities in order to identify different pathogens, right? And this is important for identifying pathogens that you haven't seen before, right? This means that your immune system, the first time it sees something, it's able to generate a response because of this great diversity of these receptors on B cells and T cells. It means that there's something out there that can more than likely bind to that pathogen, okay? And, right, that's why, right, we see some people, right, able to have an immune response to the coronavirus right away. Well, even the people that are dying, which is super sad, their immune system is responding to the virus, and it's able to do that because of this variation of receptors. And you may have heard too what they're trying to do for some folks, and I haven't read if they've actually given a patient this yet, but what we can do is, after you've been exposed to something like the coronavirus, right, and then you survive, hopefully, in your circulation, you will have antibodies that your B cells made with the help of T cells that were fighting the coronavirus. And so what they're doing now is they're taking some patients that have survived the coronavirus, they're pulling out their blood, they're isolating the antibodies, and then they'll give those antibodies to someone who has an active infection. And those antibodies will hopefully bind to the virus and aid that person's immune system and allow them to recover much more quickly and hopefully avoid severe illness. Okay. And so you can see, right, this is one of the numbers of some of the combinations. And so it's kind of like an amino acid sequence, or like our alphabet, right? You have all these different combinations and you can arrange them in so many different ways that you have almost limitless possibilities, right? And I'm not going to expect you to memorize this, but this is showing you a B cell receptor and how it's made. And so what you can see here, all right, for this B cell receptor is you have these V segments of DNA, and these V segments and these J segments are going to contribute to the structures of these antibodies. Okay. Specifically, what we're really interested in is these variable regions, right? So this light blue, this is all contributing to the variable region. And what it's telling you is that you have basically V1 through V40, and here you have J1 through J5. Okay? And what your body can do is it can splice these different combinations together. So you can put V1 with J1, V1 with J2, V1 with J3, and so on and so on. But then you can also do V2 with any of those, V3 with any of those, V4 with any of those, right? And so that gives you a very, very diverse group of B cell receptor and antibodies that are actually secreted work the same way, right? A very diverse combination and number of possibilities, right, that lead to a B cell receptor that could generate or that could respond to a pathogen. Now, truth be told, too, right? A majority of these combinations that you make will never see or respond to a pathogen. And that's fine, right? Because they are there in case something crazy and weird happens and a pathogen comes along sometime in the future or during your lifetime that would be recognized by this combination of receptors. Okay? And I don't think, yeah, we're not going to go over it here, but it's the same idea in the T cell receptor. All right, so the same basic kind of principle applies. If you are really interested in this stuff, which this stuff is super interesting, I would suggest at some point before you graduate taking immunology with Dr. Chrisman. So he's going to offer it next fall. So I don't think you guys will be ready then, but those of you that are freshmen, he'll offer that again next year, but you probably won't be ready. He'll offer that again your senior year. And so if this is something that you find really cool, I would highly suggest taking immunology with him in a couple of years. Okay. So, you know, one of the things that I've been mentioning to you guys is the fact that, you know, sometimes self-antigen is presented, and that's normal. And so your cells of the adaptive immune system have to be able to distinguish between self and non-self. Okay? And it turns out, right, that they are trained for this. So B cells and T cells, as they're maturing, either in the bone marrow or the thymus, are tested. Okay? And they are tested for their ability to respond to self-antigen. And in this case, right, this is not a good test to pass, right? So if I put a self-antigen in front of a B cell or a T cell, and it binds to it or it recognizes it, right? And this happens in the bone marrow or the thymus, that B cell or T cell will be removed from the population. Okay? So B cells or T cells that recognize self-antigen, right, either in the bone marrow or the thymus as they're maturing, are removed from the system. Okay. Let's see here. Hey, Spencer, what type of diseases or disorders arise from people whose B cells and T cells are recognizing self-antigen, right? So this control mechanism fails. So what type of disorders or diseases do those people have?
You mean like if the B and T cells aren't working?
Well, so if the B and T cells produce antibodies, right, or are recognizing self-antigen, right? They normally should be killed or removed from the system. But sometimes the system fails, and then those cells are released into circulation. So then what happens to those patients or those people that
I
have
mean,
B
like immunodeficiencies.
well, so not necessarily immunodeficiencies, close, right? Autoimmune disease, right? So immunodeficiency could be a lack of B cells and T cells. Their B cells and T cells can do their job maybe, but there's just not enough of them. So that could be a type of immunodeficiency. But, right, if B cells and T cells are released into the body that recognize self-antigen, right? So for whatever reason, and there's a number of reasons that this happens, those cells aren't removed from the system, then they will start reacting and interacting and destroying, right, host proteins. And then that is what leads to, or is one of the reasons, right, that we have autoimmune diseases, right? So my favorite autoimmune disease is type 1 diabetes. And in type 1 diabetes, B cells and T cells kill the insulin producing cells that are in the islets of type 1 diabetics. Okay. If you know someone that has MS, right? MS, in the case of MS, the immune system is attacking certain neurons in those folks, okay? And that's what causes a lot of their issues and problems, okay? So we want to remove these cells from the system as best we can, okay? So let's kind of take a step away from that and really briefly here, we'll do this slide and maybe one or two slides after it. What does it look like when a B cell or T cell is responding to a pathogen, right? So it's reacting to what it should react to. It's doing what we want it to do, right? So how does that response get amped up? How does it become effective enough to remove that pathogen? And the answer is a process called clonal selection, okay? Now, a lot of textbooks do this and focus on clonal selection of B cells, but I do want you to know that a similar process happens with T cells. And so basically clonal selection is you are taking a B cell or a T cell that is responding to a pathogen, right? And you are making a bunch of copies of it because you want it to fight that pathogen. Now, this is also where we introduce, and I've mentioned this before, but we introduce this concept of a memory cell, all right? So not only are you making a bunch of cells that can fight that pathogen, you are making a handful or a collection of cells that stay around and look for that pathogen to return, all right? And so this is really what we're trying to generate when we vaccinate you guys, right? When you get vaccinated, what you're trying to do is you're trying to produce long-lived memory cells that recognize that pathogen quickly when it returns. And it's one of the reasons that you don't even notice when you get infected or you have that infection, because it's so short-lived, because your memory cells are able to respond immediately. Okay? So this is what clonal selection looks like. And I like this because this reinforces what we talked about with B cell receptors. Remember, a B cell receptor recognizes a specific antigen. So here you see 3 different B cells, right? A B cell with a blue receptor, a purple receptor, and a green receptor. And in this case, it is the purple receptor that is recognizing the antigen. So this is the one that you want to make copies of. If the purple receptor is recognizing the antigen, but the green receptor's not and the blue receptor's not, well then why would you want to make a bunch of copies of the blue receptor and the green receptor? That is a waste of energy and resources because it doesn't help you remove this pathogen. Okay? So you make copies of this, sorry, where did that go? There we go. You make copies of the B cell that is recognizing the pathogen, all right? So this is clonal selection. So now we have a bunch of cells here, right? They're proliferating. From this group, we have something called plasma cells. Plasma cells are B cells that are producing antibodies that are now interacting with this antigen and hopefully destroying and removing that pathogen. But just as important, right, a group of these proliferating cells also becomes memory cells. And the next time you see this pathogen, your body will be able to respond quickly and remove that threat before it hopefully causes any damage to your body. Okay. And so that's what this is mentioning as a secondary immune response. Right? So the first time you ever see a pathogen, right, we call that a primary immune response. And so that takes a little longer because you basically have to go through this whole process. In the primary immune response, an antigen binds to a cell, you then have to make a bunch of copies of it, and then, right, they become these two different types. When you already have these memory cells, it's like you get to skip this step. You get to skip all this part right here. You have these memory cells, they see something binds to them, they immediately become plasma cells, and a group of them do stay memory cells, and you're able to remove that infection quickly. All right, so again, that's what a vaccine's doing. Now, in case you are wondering, right, if you think about something like the flu, well, why do I have to get vaccinated every year? Why have I gotten the flu multiple times? All right, well, what happens there, if I can go back to this slide, is that some pathogens, like the flu, are pretty smart and are pretty tricky, and they're constantly changing or evolving, all right? And so this antigen, right, may be what the flu is expressing, or the flu virus is expressing this year, but then your immune system starts to attack it, and it removes it, but, right, the flu mutates and changes, and so the next year it actually is presenting a different antigen, right? And when it does that, you have to start this whole response over again. And so this is primarily effective against pathogens that don't have a high mutation rate. As long as they're not changing, this works really well. If they're changing, right, it doesn't work as well. And so some of you probably get multiple colds a year, right? This last, or earlier in the spring semester, I had a cold, I got better, and 2 days later, I caught another cold. And that's because certain viruses, and some bacteria can do it too, are constantly changing. And so you have to start this whole process over again, all right? This is just showing you kind of the response time that it may take later. Okay, so I'll talk about this and then I'll let you go. So this is showing a primary and a secondary response. And so what this is showing, right, is that the first time you're exposed to a pathogen, right, it could take a couple weeks potentially to produce enough antibodies. So this is kind of a threshold limit, enough antibodies to fight that infection, okay? What it shows here, right, is that you have day 28, and then you can see around a little after day 35, so maybe less than a week or around a week, you get exposed to antigen A again, and you see a massive spike in the ability to produce antibodies. And this is because of those memory cells. And in this case, the response is so strong and swift that that pathogen is not able to cause illness in you, all right? But again, this is specific to antigen A. During the same time, maybe you're exposed to something from antigen B. So maybe this is one cold virus, and maybe this is another cold virus. Because it has changed and is presenting a different antigen, you have to start this response over again. And so although you feel good here and you're not getting ill from the pathogen that released this antigen, you could be getting ill from the pathogen that released this antigen, okay? So just take the time to look over that. And so we're gonna stop here. All right, we'll pick up with 43.3 on Wednesday. If you haven't yet, go ahead and look at your exam grades on Moodle. I was pretty happy across the board with them. They're pretty good test scores. And Sarah, you can back me up on there, right? They were pretty good.
Yeah, they were. I was happy with them. Okay,
good. Yeah, I glanced at them briefly this weekend, and I thought across the board it looked pretty solid. All right. So if you haven't, right, go ahead and take a look at those. We'll pick up with 43.3 on Wednesday. Please remember you have a quiz on Wednesday. And okay. So you have a quiz on Wednesday, and that should be it. All right. If you hadn't said hello, which I think you all did, make sure you do that before you leave. I'm going to close this down here in just a second. Are there any other questions, comments, or concerns? Yeah. Katie, I'll check for your test here in just a second. Okay. All right. Cool. It's good to see you guys. Hope you had a good Easter. Hope you were able to stay safe during the storms. We were lucky here in this part of Cleveland anyway. Looks like things missed us. But if you have any complications from any of that stuff, just reach out and let me know. Glad you guys are doing well though and able to be here. I miss seeing you in person, and I look forward to talking to you soonish. All right. Bye.